All right, so before we start our next bacteria, so let's just review first the basic concepts of our TSI. So in here in your screen, we have here test tubes. So let's learn how to record the result and interpret the result as well. As for your TSI, what are the three things that you need to remember? Again, for your TSI, that would be triple sugar iron. So we would be detecting number one, three types of sugar. That would be your glucose, lactose, and or sucrose. Lactose and or sucrose are usually found and located in the slant portion of your TSI, while your glucose is usually located and determined in the butt portion of the tube. So, how about your hydrogen sulfide? For hydrogen sulfide, black precipitate would actually tell you that there is a production of hydrogen sulfide. And for the gas, production of cracks and bubbles would actually signify the presence of the gas. So what we have here in this tube, so test tube number one, for test tube number one, that would be, what is the color of the tube? That would be yellow. Yellow of the slant. And for the bot, that would also be yellow. So that would only mean yellow signifies that acidity usually takes place. So if there's acidity, there is fermentation. So that would tell you in for this tube that all carbohydrates, so meaning lactose and or sucrose and glucose, are fermented. So how about the presence of hydrogen sulfide? Do we see black precipitate here? We can't see. So how about the gas? The gas, we could actually observe the presence of cracks or bubbles along the tube. So for the result, we could record the result as A over A with gas, or A over A gas positive. A over A gas positive. Why should be gas positive? Again, we could see here cracks or bubbles along the tube. A over A, that would only means acid over acid. So there's a change of color from red to yellow. How about test tube number two? For test tube number two, again, let's check for the slant. What is the color of the slant? That would be red. That would actually signifies you that the medium is alkaline. So alkaline stands for K. K. How about the bot? The bot signifies a change of color from red to yellow. Yellow signifies acidity. So that would be A. So our result would be K over A. Do we have hydrogen sulfide? Do we have black precipitate? No, we don't have any black precipitate in your second tube. Do we have cracks or bubbles? We don't have too. So our result would be K over A. What is the interpretation of this one? So the interpretation would be only glucose is fermented. Only glucose is fermented. Why only glucose? Again, glucose is found in the butt portion of the tube. And there's a change of pH of the bottom of the tube. So that would tell you that as an acidic environment would tell you that fermentation takes place. How about on your third tube? For the third tube, we have here on the slant, it's K. How about the bottom portion? That is still K. So it is obstructed. But then again, during your moving exam, you're allowed to check the setup for the tubes. So any here, so for the slant, that would be K. So for the slant, that would be K. How about the bot? If it's still red, that would be still K. So it would be K over K. So what are these black precipitate indicates? That would be hydrogen sulfide. So your result, your final result would be K over K with hydrogen sulfide, or K over K hydrogen sulfide positive. So that would be your third tube. How about the fourth tube? For the fourth tube, you could see here how the slant, that would be K over K. So there's no change of color. So K over K. So an alkaline environment would tell you that there's no fermentation takes place. That is taking place. So K over K. So no carbohydrates are fermented. No carbohydrates are fermented, meaning sucrose, lactose, and glucose are not fermented. Do we see hydrogen sulfide? There's no black precipitate, so hydrogen sulfide is negative. How about the gas? There's no bubbles or cracks, so meaning gas is also negative. Okay, so that would be your fourth tube. So let's check the next slide. So for this slide, you could actually see the first tube is the uninoculated test tube of your TSI. What do you mean by uninoculated? Meaning there's no bacteria being inoculated or strict inside the medium. So you could actually tell the difference because the second tube is the inoculated one. So again, you may check the setup during your moving exam for this one. So let's start with the second tube. How about the second tube? Since this is TSI, let's check first the slant. What is the color of the slant? That would be red. How about the color of the butt? That would be still red. So if it's red over red, what would be our result? That would be K over K, meaning alkaline over alkaline. If it's alkaline over alkaline, that only means that no carbohydrates are being fermented. No carbohydrates are fermented. Do we see a black precipitate? No, we can't see a black precipitate. Do we see cracks or bubbles? So no. So meaning hydrogen sulfide negative, gas negative. How about the third tube right here? So for the third tube, you can see again, let's check for the slant. For the slant, what's in the slant? What's the color of the slant? That would be red. How about the color of the butt? That would be yellow. So if we have a red slant and a yellow butt, that would actually tell you that the slant is alkaline and the yellow butt is acidic. So how are we going to report our result? So that would be K over A. K over A. Do we see black precipitate? No, we can't see black precipitate. So that would be negative for hydrogen sulfide, negative for hydrogen sulfide, and also negative for gas because we can't see bubbles or cracks. How about the last tube? For the last tube, let's check the slant. So in this case, the color of the slant is closer to yellow compared to red. So it's closer to yellow compared to red. So the slant should be A. How about the butt? Still yellow, so that's still A. So A over A. So A over A. Do we see black precipitate? No. So how about cracks on the tube or the medium? So we could see bubbles and cracks. So that would only indicate that it is positive for gas. So what is the result? How are we going to report the result? That would be A over A with gas, or A over A with gas positive or gas positive. How are we going to interpret? So it's A over A, meaning sucrose and or lactose and glucose are fermented with the production of gas. So again, sucrose, lactose, and glucose are fermented with the production of gas. So let's check about this one. So we have here, these are what we have been discussing a while ago. But please take note that when you report your result, do not report your result based on the color for your TSI. So in here, it was written red slant, red bot. That should be not that is not the correct way of reporting of your TSI. So it will always be alkalinity or acidity over acidity, or K over K. Okay, it could be K. For this one, it should be K over K. For the third tube, that would be K over A. So for the fourth tube, that would be A over A. That is how you interpret, how you report the result. And do not forget, if there's presence of H2S, do not forget to include that in your reporting. How about for the LIA? So the LIA in here, just like your TSI, it has a slant and a bot. What are the things that we need to remember for LIA? So if for TSI, we need to observe for the acidity. That would mean that there's fermentation going on. For the LIA, we need to check for the alkalinity. Okay, so it's alkalinity. So for the slant, we need to check for the presence of lysine deaminase. Lysine deaminase. For the bot, that would be lysine decarboxylase. Lysine decarboxylase. So for the slant, what is the positive test or positive color for the slant that would actually tell you that it is positive for lysine deamination? That would be red. Again, that would be red. So if it's red, then it is lysine deaminase positive. If How about the bot portion? How about the bot portion? That would tell you the presence of lysine decarboxylase. So it would tell you the presence of lysine decarboxylase. But then again, what's the color of the bot? That should be purple. Okay, that should be purple. So how about test tube number one? So for test tube number one, we could observe first for the slant. What is the color of the slant? That is purple. Okay, so the color of the slant right there, it's purple. So meaning lysine deaminase, is it positive or negative? It's lysine deaminase negative. How about the color of the butt? The color of the butt is purple. That would tell you that lysine decarboxylase is positive. So how are we going to record the result? That would be K over K. Why K? Because we are actually checking again for alkalinity. K stands for alkaline. So K over K. So K over K, do we see gas? We can't see gas. Do we see the presence of hydrogen sulfide? We can't see the presence of hydrogen sulfide because we don't have any black precipitate along of your tube. So the result should be K over K. How are we going to interpret this one? Lysine deaminase negative, lysine decarboxylase positive. Okay, that is for your test tube number one. How about test tube number two? For test tube number two, we could see here what is the color of the slant for test tube number two? The color of the slant of test tube number two is red. The color, as you can see here, and the slant, it's red. So red indicates the presence of lysine deaminase. Take note, on the slant, lysine deaminase should be identified. So lysine deaminase slant, the positive color should be red. So for test tube number two, lysine deaminase is positive. How about the bot portion? The bot portion of our test tube number two is yellow. It is yellow. So yellow indicates fermentation since it's acid. Glucose fermentation is positive for glucose fermentation, but it is negative for lysine decarboxylation. Why is that so? Again, for lysine decarboxylation, the positive result should be purple bot. Purple bot. For in here in test tube number two, you could see here a yellow bot. Yellow bot. So what is the result for test tube number two? How are we going to type our result? The result should be what is the color for test tube number two. This time you could actually write the color. It should be R, that stands for red. What is the result for the bot? It stands for A, means acid. R over A. R over A. So R means lysine deaminase. What is the interpretation? Lysine deaminase positive. Lysine decarboxylase negative, but with glucose fermentation. Again, for test tube number two, our result or our interpretation should be lysine deaminase positive, lysine decarboxylase negative with glucose fermentation. Okay, that is how are we going to report our result for Leah. Okay, so these are the right one. So these are what I have been saying a while ago. All right, so we're done with E. coli. So let's go with the primary intestinal pathogens. So let's start. So we have Salmonella, Shigella, and Yersinia, but don't you worry. These three groups would be the coverage or shorter compared to your E. coli. So let's start with Salmonella. So I actually have here a video. So I will not show you here lang in this pre-recorded video. I will be uploading this video on your Batch G drive, so you could actually tell. That is a fun way of learning or remembering who Mary Mallon is and who Mary Mallon is, and what is the importance of Salmonella. Okay, so let's start with Salmonella. Specifically, we'll focus on Salmonella typhi, Salmonella paratyphi, which are both responsible for enteric fever and acute gastroenteritis. So let's start with its general characteristics. What is enteric fever, by the way? So enteric fever is also known as the typhoid fever. So obviously, it is caused by Salmonella typhi. So it is caused by Salmonella typhi. So how are we going to get this bacteria? So the disease results from the bacteria that invades our bloodstream. So this disease usually happens or starts when it invades our bloodstream. That is why it's fever. So we're talking about systemic symptoms. So if there's fever again, there is systemic symptoms. So it actually starts with abdominal pain because we could you could actually get this through ingestion of contaminated food. So it starts with abdominal pain, cramps, diarrhea, and then it may go further from systemic symptoms or prolonged fever. That is why it is known as the enteric fever. Enteric means the causative or the culprit of this fever are the enterics or the bacteria that are usually reside on the enterocytes or the intestines. So one example is your salmonella. Just remember your salmonella, shigella, and yersinia, unlike your E. coli, these microorganisms are considered true pathogens, meaning they are not normally found inside our body. So it should never be found inside our body. The moment that they're inside our body, that is also the same moment that they would cause infection. So what we have here is acute gastroenteritis. Another one is acute gastroenteritis. So this condition is often associated with foodborne infection or intoxication. So meaning intoxication, it usually causes by toxins or mediated by toxins. And it is very common to Salmonella species as well. So aside from the typhoid fever, um, Salmonella also causes gastroenteritis that includes symptoms of diarrhea, vomiting, abdominal cramps as well. So, unlike other Salmonella species, Salmonella typhi and Salmonella paratyphi do not have animal reservoirs. So, they do not have animal reservoirs and they are exclusively human pathogens, meaning they cannot infect animals. So even if you have your typhoid fever and you have a pet dog, your dog cannot get the Salmonella typhi. So it is exclusive to humans. So it is classified as exclusively human pathogen. Meaning, if it is exclusive to human pathogen, the transmission is high between two people. So, meaning they are actually transmitted only from person to person. Okay, so they are transmitted from person to person. So, how are we going to get this one? So, usually the habitat, these bacteria primarily inhabit the gastrointestinal tract. That is why it causes gastroenteritis, right? So, usually it inhabits the human gastrointestinal tract, and the main mode of transmission is through, again, the ingestion of contaminated food and products, such as improperly cooked poultry or milk or eggs, usually dairy products. Um, Human carriers are also very common of salmonella. So when you say carriers, we have already discussed this in our parasitology class that when it is a carrier, these are people who are carrying the bacteria but do not manifest symptoms. They do not manifest signs and symptoms. So they're carrying the bacteria without manifesting such symptoms. One example of a human carrier is Mary Mallon. So that is the video that I will be showing you supposedly. So I'll be uploading that in the Batch G drive. So Mary Mallon is a very historical case. It She was referred as Typhoid Mary. So why Typhoid Mary? Because she was a cook long time ago in the early 1900s. So she unknowingly transmitted typhoid fever to 51 individuals. Imagine that. So in that video, you would learn na Mary Mallon transferred from one place to another. At first, she didn't know that she was actually transmitting the disease, but later on, she was chased by police officers and healthcare investigators because she was later known to be the culprit of such disease or infection. So, why do we have these career state? Class for Salmonella, salmonella, the organ for chronic carriage is the gallbladder. So the gallbladder is the site of chronic carriage, meaning individuals who are chronic carriers can harbor Salmonella typhi or paratyphi without showing such symptoms. But then again, antimicrobial therapy can actually help terminate the carrier state. But what is very unique of these bacteria is there is no evident gallbladder infection. So there is no evident gallbladder infection. So meaning the bacteria would actually harbors and stays in the gallbladders, staying there and causing infection, and it could actually could help in the transmission of the disease. So if the gallbladder is infected, the moment that the gallbladder is infected, surgical removal of the gallbladder may be the only effective solution to end the chronic state. So say for example, if person A is actually identified as a chronic career, no? So this person A will be treated with antimicrobials or antimicrobial therapy or antibiotics. Say, for example, antibiotics won't work for this person. So this person or person A would be subjected if he or she wants to end his or her chronic state. So he or she would be subjected to cholecystectomy. What is that one? That is the surgical removal of the gallbladder. So tangtangon iyang gallbladder just to end the chronic state. So this is actually a very good discussion, a very good highlight or emphasis of good or importance of food safety and proper cooking practices, especially when we are actually handling dairy products because salmonella is very common to dairy products. Okay, so much more for the disease. So let's talk about the general characteristics of salmonella. Again, this is an enteric type of bacteria, so meaning it is still gram-negative, rod-shaped bacilli. So rod-shaped bacilli, as you can see there in your screens, we have here bacilli. Dagko kay sila, no? So that would be rod-shaped bacilli. It is aerobic or facultatively anaerobic, meaning these bacteria are very flexible in their oxygen requirements. They can thrive both in the presence and the absence of oxygen. That is why they are classified as facultatively anaerobes. But then again, when you work in the hospital lab, in the hospital setting, it is much better. So mas guwapa gid sila, mo grow jud sila better with the presence of oxygen. But by the book, it can grow both with the presence or without the presence of oxygen. It is lactose negative. So Salmonella species are non-lactose fermenters with the exception of one species. What is that? If you could remember, it is a late lactose fermenter. That is Salmonella arizinae. It is Salmonella arizinae. And it is also motile. So it has a peritrichous flagella. So they are equipped with this peritrichous flagella distributed all over the body. So as you can see there in the picture, the upper part of your screen. So that is an example of a peritrichous flagella, like this one. So we have also exceptions of some of the salmonella species. Most of the salmonella species are motile, but we have also certain bacteria or species under salmonella that are non-motile, and that is Gallinarum and Pelorum. So we have also here that salmonella is usually generally they are uncapsulated. They are uncapsulated except with Salmonella typhi. So generally again, Salmonella species do not have capsule. If you could remember the mnemonics, so some killers have nice capsules. So generally they are not part of those killers. So they are uncapsulated. However, Salmonella typhi is an exception. It has a capsule that plays a role in its pathogenicity. So if there's a capsule, you already know that it usually evades phagocytosis. We have here the virulence factors. For the virulence factors, just like your E. coli, it also has a fimbriae. If you remember what fimbriae is, so these are your hair-like projections on the bacterial surface. So these are small hair-like projections, which facilitates attachment or adhesion. So therefore, your salmonella also has this fimbriae. So it usually attaches to your intestinal lining, and that could promote or improve the production of improve the manifestation of diarrheal symptoms. So that is the purpose of your fimbriae. So through this fimbriae, it could actually establish the infection more effectively. Meaning, so even if you drink water a lot, no, kung sikag inom tubig, the bacteria could resist being flushed out by peristalsis. So it is not effective because of your fimbriae. So the only treatment for this bacteria is your antimicrobials. So we have also exotoxins. So Salmonella produce exotoxins, so they are toxin mediated. So these same toxins can damage host tissues. So they damage host tissues, and you could later on you would see that it would actually contribute to the disease process itself. So other than that, we have here some antigenic structures. So if you could remember the somatic O antigens, so somatic O antigens, those are the integral components of your lipopolysaccharides, meaning, just a review, your O antigens are important for serotyping. So they're important for serotyping or the differentiation of the different variants of the bacteria. How about the flagellar or H antigens? So your flagellar or H antigens, if you could remember, salmonella generally are motile. So these are actually found in the flagella. So if you have flagellar or H antigens, this would actually tell you that salmonella is motile since these antigens are responsible for motility. How about this next antigen? The VI surface antigen. If you could remember, your VI surface antigen is a subclassification of your capsule antigen. So VI surface antigen is a capsule antigen. It is also it acts a very as a virulence factor. So if your salmonella is capsulated, right? As you can see here, most of your salmonella is uncapsulated except your salmonella typhi. So if it has a capsule, the name of its surface antigen is your VI antigen. And again, if the bacteria has a capsule, it is used for phago or inhibiting phagocytosis. It is used against phagocytosis. All right, so what we have here are the general characteristics in identifying Salmonella species. So these are some of the common plates that you should be using if you suspect Salmonella on your clinical specimen. So number one, did you na makalimtan? It's a staple agar for enterics. That would be Makongke. So if you could remember your Salmonella species, generally there are non-lactose fermenters except your Salmonella arizanae. So your Salmonella, generally Salmonella species has clear and colorless colonies since they are non-lactose fermenters. Okay? Since Salmonella does not ferment lactose, which means it doesn't change the color of the media. So it will not turn pink except for arizona. How about this media? So your media right here are media with hydrogen sulfide indicators. So your salmonella is known producer for hydrogen sulfide. So it is a known producer for hydrogen sulfide. Number one, what we have here is your HEA, your Hectoin enteric agar. In here later on, I will show you pictures. In here, Salmonella colonies are green with black centers. Black centers usually indicates the hydrogen sulfide production. So the black centers indicates the production of your hydrogen sulfide. Why is it that there are black centers? So these black centers are actually the reaction of your iron salts. So the iron salts will react to the hydrogen sulfide, producing black centers. And it is also true to your XLD. What is your XLD? Your XLD stands for xylose-lysine deoxycholate. Like xylose-lysine deoxycholate. So on your XLD agar, Salmonella colonies produce red colonies with black centers. So if your XLD right here, if your XLD produces green colonies, so if your HEA produces green colonies, your XLD produces red colonies. Your XLD produces red colonies. So still with black centers. So your black centers would actually indicate that the bacteria has a capacity or ability to produce hydrogen sulfide. Hydrogen sulfide. Why is this red? So the red color indicates of your XLD. So it's red colonies now. Why is this red? Because these red colors would actually indicate that your salmonella can ferment xylose. Xylose is a type of sugar. So it is red because your salmonella can ferment xylose. So it has red colonies with black centers. How about your bismuth sulfite agar or your BSA? So this medium is particularly useful for isolating Salmonella typhi. So colonies usually appear metallic with a ring around them. So later on, I will show you an example. So this metallic sheen, if you could remember your E. coli, so that was a greenish metallic sheen. This one is just like a metallic sheen. So a metallic sheen actually confirmed the presence of S. typhi. This is a very unique feature of Salmonella typhi. Again, BSA Salmonella typhi metallic ring. So how about Salmonella Shigella agar? So Salmonella Shigella agar is also known as SSA. So SSA Salmonella colonies, it is used to differentiate Salmonella versus Shigella. On Salmonella colonies, the colonies are colorless but with a black center. So again, the black center indicates hydrogen sulfide. So that is for Salmonella. Okay, so that is for Salmonella. The colorless appearance of your Salmonella would distinguish Salmonella from other bacteria that might produce colored colonies. But for your Shigella later on, you would know that there are no black center because your Shigella is not a hydrogen sulfide producer. So it cannot produce hydrogen sulfide. So in your SSA, colorless colonies with black center, that would be your Salmonella. So what we have here is an example of your Maconke. Again, for your Maconke, for your Salmonella are known to be, generally, they're known to be non-lactose fermenters. So this is an example of your non-lactose fermenter. As you can see here, these are all the comparison of lactose fermenters and non-lactose fermenters. On the middle part of your screen, we have your Salmonella typhimurium. So Salmonella typhimurium, if inoculated in your macunque, shows clear and colorless colonies. You could actually compare that with your E. coli at the upper left of your screen and your E. aerogenes on the upper right of your screen. If you could remember, E. coli and Enterobacter aerogenes are rapid lactose fermenters, while your Salmonella, on the other hand, are non-lactose fermenter. Salmonella, except your Salmonella aeruginoe, which is a late lactose fermenter. So same concept class. If it's non-lactose fermenter, colorless colonies. So this is what I have been talking about. So we have your Hectoin enteric agar urhea. You could see the difference between your Shigella and Salmonella. For your Shigella flexneri on your left portion, you could see colorless colonies but without the black center. Again, your Shigella is a non-hydrogen sulfide producer. So it cannot produce hydrogen sulfide. Unlike your Salmonella on the right portion, it could produce hydrogen sulfide. So you could see here colonies with black centers. So your clue for Salmonella, black centers. No, if there are black centers, think of Salmonella. Also the same with your XLD. XLD, your XLD stands for, what does it stand for? Stands for again, xylose lysine deoxycholate. So that would be xylose lysine deoxycholate. So you can see here growth on your Shigella, both of your Shigella and Salmonella. But then again, what differs Salmonella from your Shigella? You could see Salmonella colonies are typically red with black centers. Why again? Because Salmonella, on the other hand, ferments lactose and could also produce hydrogen sulfide. That is why the red color colony of your Salmonella is because of the fermentation of the xylose, while the black center indicates right here the hydrogen sulfide production. How about Shigella? Your Shigella again does not produce hydrogen sulfide. It does not produce hydrogen sulfide, so there's no black center. It also does not ferment xylose in a manner that it would really produce red colonies. No? So dili gid siya ing anak ka ferment. Dili gid siya nga strong in fermentation na the colonies would really turn to red. So what we have here, we could observe yellowish to colorless. That is due to the lack of fermentation of xylose. So I'm not saying that Shigella cannot ferment xylose. It's just that your Salmonella is a strong fermenter of xylose compared to your Shigella. So as you can see here, Shigella, it's yellow or colorless. Okay, it's yellow or colorless. But what is very important right here is the production of your hydrogen sulfide. The production of your hydrogen sulfide. So your hydrogen sulfide production, meaning there's no black center. Okay, so let's proceed to the next. So there are different ways of salmonella that can make us sick. So each ways or each step has different symptoms and causes. So a while ago, I have already introduced to you the enteric fever, the gastroenteritis, and then the bacteremia. I haven't introduced the bacteremia a while ago, but you are already aware what bacteremia is. Okay, so if worst comes to worst, no, all of the bacteria would actually cause bacteremia or sepsis if not treated. Excuse me. Okay, so what we have here, enteric fever, also known as your, if you could remember, that is your typhoid fever. If you could remember typhoid fever, since it has fever, it is already a systemic infection. What is the most common culprit of your enteric fever or typhoid fever? That would be Salmonella typhi. So that would be Salmonella typhi. So how are we going to get this enteric fever? That is actually transmitted through ingestion of contaminated food or water. So the moment that the food or water, so the bacteria could actually invade the intestinal lining and could actually enter the bloodstream and could spread throughout the body. And that is the moment wherein we experience typhoid fever. So it goes beyond diarrheic symptoms. Now it goes beyond the diarrheic symptoms. It becomes systemic. So that is why it is enteric fever or typhoid fever. How about gastroenteritis? What is gastroenteritis? Gastroenteritis. So that is the infection of your gastrointestinal tract. Itis, meaning inflammation, means infection. So that it actually involves both the stomach and the intestines. So very common to enteritis and typhimurium for gastroenteritis. So it is also associated with foodborne outbreaks or foodborne illness. How about bacteremia? So bacteremia usually happens when Salmonella bacteria enter the bloodstream and it could actually spread throughout the body. So this could be a complication of your enteric fever. Remember what I told you that all diseases, if not treated, would be the result or would be the sequelae would be bacteremia or a complication will happen and that complication would be bacteremia. Why? Because our immune system is already weakened. So this may include fever or chills, and maybe signs of sepsis. So it could actually lead to some serious conditions, and patient may also experience septic shock if not promptly treated. So these are the things that I have been telling you a while ago. So these are enteric fever. So the main way, again, through how do we the main mode of transmission or the main acquisition of enteric fever, or how the bacterium gets into our bodies, the main way is through eating or drinking contaminated food or water. So just imagine you eat something that's been touched by an infected person, or you drink water that wasn't properly treated. It is also very possible that you get this from surfaces or objects that have been contaminated by someone carrying the bacteria. So, as what I have been discussing a while ago, your Salmonella typhi or your typhoid fever can be transmitted from person to person. It has no animal reservoir. So, meaning the level of infectivity is actually high. So, you could actually get this through through people, so from other infected person. So this is how the your typhoid fever looks like. So your typhoid fever, a notable sign to look out for is the rose spots right here. So the rose spots are small, as you can see in this picture, small reddish marks that usually appears on your stomach and chest. And it is very common during the second week of fever. That is the unique characteristic of typhoid fever. So aside from the usual tiredness, muscle aches like malaise, myalgia, lethargy, so feeling very tired, not having much appetite. So experiencing muscle pains or aches. So those are some of the common symptoms of systemic infection. You don't have appetite, you feel very tired, you feel muscle aches everywhere. So you have persistent headache. So that is a systemic infection symptom. Aside from that, a notable sign to look out for is this one, the rose spots, which happens during second week of fever. So there is actually a complication. So if if enteric fever is not treated, it can lead to some serious complications. And an example of which is your Peyer's patches. So Peyer's patches is a lymphatic tissue in your intestines, and it can actually cause necrotizing cholecystitis and Peyer's patches. So these are more severe digestive problems. So meaning when you say cholecystitis, that is the inflammation of the gallbladder. No, so Peyer's patches, that would refer to the lymphatic tissues in your intestines if you remembered your histology class. So, how do we get this one, and why do we experience diarrhea? So, here's a bit of how it works. So, after you ingest the bacteria, Salmonella typhi, you need to remember that it is an enteric. So, it attacks first the lining of your intestines. So, it then moves to the nearby lymph nodes. So, it doesn't stop there on your intestines. It attacks, it adheres, it invades. After invading, it moves to the lymph nodes and spreads throughout your bloodstream. So, this bacteria is very unique because take note, it can actually hide inside the immune cells. Asa man sila mo hide? They would hide in the immune cells called the monocytes. So, if they hide inside the monocytes, it would actually help them evade the destruction of some phagocytes. Ano, kung matago man sila sa monocytes. So, a monocyte is an immune cells. So, they will hide inside the monocytes, and it would make the infection harder to treat. So, that is one of the unique pathophysiology of your salmonella. So, what we have here is gastroenteritis. Please take note that gastroenteritis can be caused by several types of salmonella. But what is very common is your Salmonella enterica, subspecies enterica. So if you could remember that one, so your Salmonella enterica, subspecies enterica, that is a specific serotype of Salmonella. So another one is your Salmonella typhimurium, which is also linked to outbreaks of gastroenteritis. So how does this bacteria make its way into our system? Again, one major route is through the use of contaminated cooking utensils or eating these contaminated food. So through the food, the bacteria may spread inside our body. Another one is inadequate refrigeration. So if you do not refrigerate or store your food properly, it could actually contribute for the bacteria to multiply and, and grow more rapidly. So the sources of infection usually in this bacteria is the poultry, dairy products, and even the handling of pets. So sorry. So if you're familiar with the peanut butter issue, so the peanut butter issue, the Jif peanut butter outbreak. So to illustrate how serious these infections can be, in recent years, there was a significant recall of Jif peanut butter, the JIF. I don't know if you're familiar with that, but that brand of peanut butter was um, claimed back because of a possible contamination with salmonella. So, but then again, it came too late. The news came too late. Most of the people already bought the Jif because Jif is Koanman is famous in the United States. So this incident actually led to numerous cases of gastroenteritis, and it actually highlights the importance of, aside from importance of remembering or taking notes of the expiry date, it also highlights how widespread and impactful such outbreaks can be, because most of the people who bought Jif suffers from severe gastroenteritis. So we have here some certain types or brands of peanut butter if you're fond of eating peanut butter. But then again, there's also an issue about Skippy, but it was later on settled. So we have here, I have posted a picture of CBC. Why CBC? Because there are our superheroes. So they're actually the one who would monitor food safety and some outbreaks. So we could actually CBC could actually help us, in preventing these kind of infections. So bacteremia. So bacteremia is a common term that you would hear over and over again in the bacteriology class. So bacteremia is a condition where bacteria enters, and it doesn't just enter, but again it circulates in the bloodstream. So it invades the bloodstream and circulates in the bloodstream. So in the context of salmonella infection, this could actually occur with or without additional foci of infection. What do you mean by foci? So even if you suffered from gastrointestinal infection. So it doesn't mean if you have suffered gastrointestinal infection, it will just end as a gastrointestinal infection. So it would go further and progress further to become bacteremia. So it is quite possible. So we have also some non-typhoidal strains of salmonella right here that can lead to bacteremia. So meaning we have non-typhoidal salmonella. So those salmonella that cannot cause typhoid fever but could cause bacteremia. So that is presented in front of you. So what we have here is the characteristics of your bacteremia. So we have your prolonged fever. So intermittent bacteremia. What do you mean by intermittent again? On and off. So meaning this, the presence of bacteria in the bloodstream can vary. Sometimes it is detectable and sometimes it's not. So that is why it actually makes the diagnosis and monitoring of salmonella infection complex and difficult. But then again, that is the job of the doctors. So again, for bacteremia, it is also common to non-typhoidal salmonella. Please take note. So what we have here are some salmonella species, and each salmonella species has subspecies. And we have here the most important species that has three serotypes. Okay, so let's just introduce. Let's start first with this genus and specific epithet. So we have Salmonella and Enterica. Salmonella enterica, that would be your species for Salmonella, very common species of Salmonella. Salmonella enterica and Salmonella bongori. So your Salmonella enterica, this one, is subdivided into 6 subspecies. So what are these subspecies? We have Salmonella enterica subspecies enterica, Salmonella enterica subspecies salame, Salmonella enterica subspecies arizona. We have diarizona, hotene, and indica. So, question: among all of these subspecies, which of these subspecies is a late lactose fermenter? That would be your Salmonella enterica subspecies arizona. So, we have also another subspecies. So we have here the Salmonella enterica subspecies enterica. So your subspecies enterica are divided again into 3 serotypes. So how are we going to write this one? So you are going to write Salmonella enterica subspecies enterica serotype typhi. We have Salmonella enterica subspecies enterica serotype choleraceus. We have Salmonella enterica subspecies enterica serotype paratyphi. Therefore, Salmonella enterica subspecies enterica serotype typhi, choleraceus, and paratyphi are the most clinically significant serotypes under Salmonella. Okay? So those are the 3 most clinically significant under Salmonella. Okay, let's proceed with the identification of your salmonella. So if the IMVIC reaction of your E. coli is positive, positive, negative, negative, please remember that in your salmonella, that would be positive, negative, positive. So for your serotype, that would be, for your serotype type V, that would be negative, positive, negative, negative. So please take note that all salmonella species are motile. So they are motile with the exception of Salmonella pullorum and Gallinorum. So therefore, pullorum and Gallinorum do not have your H antigen. So again, these serotypes do not produce gas, but then again, your Salmonella serotypes, most of them are active producers of hydrogen sulfide. Hydrogen sulfide with the exception of Salmonella serotype paratyphi So paratyphi So your Salmonella again is has a very specific pattern for your INVIC and also for your TSI. For your TSI, it is K over A. That would be alkaline over acid, K over A with gas production with hydrogen sulfide. K over A with gas with hydrogen sulfide. So except langni, Salmonella serotyphi, which cannot produce gas. Again, which cannot produce gas. Imong hydrogen, your Salmonella serotype typhi. But again, generally, the TSI reaction of your Salmonella is K over A with gas with H2S. So this is how are you going to differentiate your Salmonella species. Usually, they are differentiated with your TSI reaction. So V stands for variable. So V stands for variable, meaning it would vary its result. So it would your Salmonella can be again differentiated based on your TSI and your IMVIC, and that also includes the sugars. So these are some of the sugars and some enzymes that could help you differentiate one serotype from the other. So to diagnose salmonella infections effectively, we collect different specimens at various stages of the infection. So here's how it typically works. So for the first week of infection, that would be the blood specimen. So why is it the blood? Blood is the primary specimen collected during these early stages of infection because salmonella bacteria can be easily detected in the bloodstream. So at this first week of infection. And how are we going to detect that one? So the doctors would initiate in requesting blood culture. So blood cultures are very critical for identifying bacteremia and diagnosing the enteric fever or typhoid fever. That is why during the first week of infection, blood is the primary specimen collected during these early stages. Okay, so during this period, we can actually detect salmonella in the bloodstream already. How about the stool specimen? So the stool is the timing should be the second week of infection. So class as the infection progresses, especially in the second week, salmonella are more likely be present in the gastrointestinal tract. So stool specimens are collected to identify the bacteria in the feces and helping to diagnose some diseases like your gastroenteritis. And this specimen could also help us track the infections of the progression. So the progression of the infection. Okay. So that would be your stool. So we have here the urine specimen. For the urine specimen, that would be the third week of infection. So by the third week of infection, the bacteria may spread to the urinary tract. So urine specimens are actually collected to check for the presence of salmonella in the urine. So if there is already salmonella in the urine, this can be indicative of a systemic infection or some complications of the bacteria, such as sepsis or bacteremia. So that is why this is actually the um, various stages. So we collect different specimens at various stages. So just remember your BSU, blood, stool, and urine, first, second, and third. Okay, so for the Shigella species, I will end my lecture video here and then I would record another video for your Shigella and Yersinia.